Okay guys, let's start our afternoon session. So now I can tell you that whole story that I was waiting for. Okay. Now, I showed you how NO releasing materials are great. They can kill bacteria in that CDC bioreactor. They can prevent clotting. I'll show you in future. One problem is they cannot prevent protein absorption. Okay, so if you have an implant, fibrinogen, protein, it's still sticking to the surface. So my lab over the years is working on lot of different anti-fouling strategies also. Okay, we think, so our goal is to go as close to endothelium as possible, the blood vessel that's carrying blood. Nitric oxide is only one of the mechanisms. We believe that in that final material that we want, nitric oxide will have one role. But we also know we need something more. Okay. And now we want, can we make a surface anti-fouling and nitric oxide releasing? So, if anything, protein, bacteria comes, this anti-fouling surface will prevent protein absorption, and nitric oxide will prevent platelet activation and bacterial contamination. Right? So, over the years, we have published a lot of papers that you can actually, if you are interested, message me, I can send them to you, where we have combined NO with slippery surface. That's what I'm going to talk today. NO with super hydrophobic surface, super hydrophilic surface, Zwitter ion surface, or conjugation of bioactive agents such as heparin. All have nitric oxide, but we have tried different things with nitric oxide to see what is giving us the best synergy. I'll show you one example. Over the years, what happened? So Marcus, my first PhD student, Joanna Eisenberg is a big name in our field, biomaterials and medical devices. Her group published a paper on slippery surfaces in 2011, 2012, around that time. It doesn't say. So the idea is you have silicon oil embedded into the walls of the tubing of medical devices, and tubing is slowly releasing that oil, and that oil is making the surface so slippery that anything like blood or bacteria or proteins are just slipping off the surface. They are not sticking to the surface. So surface is anti-fouling. So I talked that why can't we make nitric oxide releasing slippery surface? Because slippery surface is only preventing things to stick to it. They are not preventing platelet activation, and they are not killing bacteria. So Marcus was the first person who started doing this work, and his paper was published in Nature Scientific Reports. So basically, this got a little bit messed up. So this is a control silicon rubber. SR is silicon rubber. NO is nitric oxide releasing surface. Li is liquid infused surface. Linorel is liquid infused nitric oxide releasing surface. And all this fluorescently labeled protein is fibrinogen. So on a silicon rubber, lot of protein sticking. NO surface, protein sticking. Liquid infused surface, protein is not sticking, and linorel surface, protein is not sticking. So we made an anti-fouling NO release surface. We also did a 7-day bioreactor study where we showed how for Staph aureus the combination of linorel and compared to only liquid infused or only NO was significantly better than by themselves. We did NO release and we found out that our NO release is pretty consistent over 60-day period. Only NO or NO plus liquid infusion. So liquid infusion is not hurting NO release. Of course, that's what we want, right? But you can also imagine that how we would be thinking when we were designing this surface, right? The question is, do we make NO releasing surface first and then we add slipperiness to it, or do we make slippery surface first and then make it NO releasing? Right? So all this is more optimization. The beauty of slippery surface is you can just swell silicon oil into the walls, just like we can swell NO donor into the walls. You know, we talked about how you can make a solution of NO donor, put your polymer in it overnight, it swells inside, and then you let the solvent come dry. Similarly, we can put it in silicon oil can go into the walls, and then it very slowly keeps replenishing on the surface, so your surface stays slippery. So you know, this fluctuation you are a little bit seeing, it's not too much. I mean, see, if you see y-axis, it's 0.4 to 0.8 or something like that. So it's not, that fluctuation is, so you know, we have 5 nitric oxide analyzers. It's instrument to instrument difference little bit, but it's not too much. Standard deviations are very tight and it's almost the same range of NO release. So then what we did, we bought some urinary catheters from market that are used in patients, and we made them, so we bought silver urinary catheters, control urinary catheters, NO releasing urinary catheters that snap, and slippery surface with NO releasing urinary catheter. And when we did a 7-day bioreactor study, we saw that our catheter was almost 2-3 log reduction better than only silver or only snap. The combination was better than anything else. We also looked under SCM, and you can see all this is E. coli. They are like rod shaped, and our slippery surface was totally almost clean. If it was not slippery surface, what would have happened? If it was just NO releasing surface. Second image is NO plus slippery. That's good, but with SCM we cannot see dead cells. I mean, you might see lot of bacteria on the surface, but if you do live dead staining, you would have seen lot of dead cells, but here you wouldn't even be able to tell that there are a lot of dead cells. You would see lot of bacteria stuck to the surface, but you don't know anything else. Megan finished her PhD 3 years ago. She did, so see, I'm showing you, we started working on this technology in 2018 or 18, and how over the years we progressed with this particular technology. We got some clinical strains of different bacteria from CDC. Some were strains, functions, and I'm just showing one here, but she tested like 6-7 strains of bacteria. Because we had a grant from CDC, they wanted 30-day studies with all those strains from CDC for us to show that our material works. These were very difficult studies because it was a 30-day bioreactor. Bacteria was growing like crazy sometimes, so she had to repeat some studies because of contamination issue, but basically we showed that we were doing very well with our material. From yesterday's class, who can tell me why do we need rabbit model, thrombogenicity model. Why can't we just do in vitro studies? Why do we need animal studies? Easy to handle, inexpensive, and you cannot do more than 2-3 hour study, and we can do longer term study, right? So yes, we did this. Then my PhD student Divine, he graduated 2 years ago. He tested those materials in extracorporeal circulation model. Okay, we little bit talked about it yesterday. So what we do is we take this tubing and we coat it from tip to tip inside the tubing with our NO releasing material. For example, if we were using the diazonium dilate chemistry, we would have done something like this. We would have first coated it with our diazonium dilate material and then top coated it with just the polymer. Now we were doing slippery surface, so we swelled the tube with SNAP solution first, NO donor, and then we swelled it with the oil to make the surface slippery. I mean, we filled the solution inside and then locked it, and then overnight or whatever time, then we filled the oil inside. Oil and NO donors went into the walls. So actually I don't have a picture of the tubing. The tubing looks green after NO is loaded in it. And we load oil in it also. So now your surface is NO releasing and it's slippery. No, because the powder, that NO donor is green color, green in color. Let me go a little bit back for this study. Few years ago in 2014, first we wanted to see, forget NO donor, we wanted to see what is the best polymer that is most hemocompatible by itself. So we did a 4-hour ECC study where we saw that E2AS polymer, that is a copolymer of silicon rubber and a polyurethane, was the best. The reason is, this is platelet count every hour. I told you in that ECC model, every hour we draw blood and we count platelets. So when we did every hour platelet count, we saw this E2AS material maintained the platelet count the best. Yes, it dropped from to 60% at 4 hours, the blue one, but it was better than other 3-4 polymers that we tested. Okay, then I also told you in that ECC model that at the end of the 4-hour study, we look at the clot formation in the chamber. So we found out that clot formed on E2AS was the smallest out of the 4 polymers. Plus, 80% of the circuits survived in E2AS, whereas only 25 to 50% circuits survived in 4 hours. Meaning, I also told you yesterday, one of the things we do with this circuit is we measure the flow rate over 4-hour period, right? So we measure flow rate, we count platelets every hour, we look at the clot formation in the end, and we also do aggregometry every hour to make sure that platelets are aggregating. That I didn't show here, but for PVC, SP60D60, SG80A, almost 25 to 50% of the circuits survived, meaning 75 to 50% circuit clocked before 4 hours. So the flow rate actually dropped to 0 in 2 hours or 3 hours. Only E2AS had flow rate of above 10 or 20 or 40, whatever it was, I don't remember now, and was keep going for full 4 hours. And remember, there is no given, there is no material yet. This was just a study to understand what polymer is good by itself. And there was a paper we saw that said that actually E2AS polymer binds more to albumin than fibrinogen. And maybe that's the reason why it was superior in properties, because albumin pacificates the surface, because it does not have that RGD peptide that fibrinogen has, which platelets and bacteria like to stick to. Okay, now going back to that slippery NO surface, something happened with all my figures. That silicon rubbers R went, that linorels L went. So I don't know, something happened, sorry. So what we did here, we tested silicon rubber tubing, just the tubing by itself, nitric oxide tubing, NO releasing, liquid infused tubing, and linorel or liquid infused nitric oxide releasing tubing in a 4-hour rabbit model. By itself, if you see this graph, silicon rubber tubing, only 65-60% of the tubing survived. Like by 2 hours, some of the loops actually clotted. The flow rate went to 0. But NO, liquid infused NO surface, they all survived full 4 hours at least. But then when we looked at the thrombus area, you can see how the thrombus area was. And when you see on the right side, you can see how there was so much thrombus in the top one, silicon rubber, and how NO, liquid infused, linorel, they all were nice. And liquid infused was maybe better than the other two. Then we wanted to test it in a longer term model. Our intravascular model, we little bit talked about it yesterday also, and also that how infection can happen in that area at the hub, clots can happen, infection can happen. And who can tell me what all we do in this model? Okay, so I want to clarify one thing. We are not doing any lock solution. So lock solution is one strategy. Like people can fill this catheter with a lock, antimicrobial lock or heparin lock, and that's fighting infection. This model is we implant the catheter in the animal for whatever time. Best I think we have done is 1 month. Now every day we can go to that hub and try to draw blood and infuse medicine or buffer or saline to make sure that the catheter is not clogged. Also we can do is at the end of the study, we look at the clotting of the catheter with the imaging and SEM, and we can take pieces of the catheter to look at the infection of different places in the catheter. Okay, one question, how do you think we can take the catheter, how do you think we take the catheter out? So we implanted the catheter in the rabbit. After 1 month, I want to take the catheter out. How would we take it out? Surgery, sure. Then what will we do? Yeah, I mean, yes. So surgery, so yes, it's 1 month now. I want to take the catheter out from the rabbit. Can I just pull the catheter out? Okay, why no? So many people said no.
Catheter out, whatever is sticking on the surface of the catheter, that might be disturbed.
Great.
So yes. First, I think we need to sacrifice the animal, then take out the entire blood vessel that we have inserted our catheter in.
Very good.
And then in sterile condition,
yes. So what we do is we cut the blood vessel very nicely, and then we take the catheter out. Because if we pull it out, if there was a clot or whatever, it'll get stuck inside, and we might see very clean catheter. See, this is what I was saying. I have some slides that are repeated. Now slide number 74. Great. Okay, so now we did this another interesting study. My student Elaine, I have her picture maybe later somewhere. She's defending her PhD on March 11th. So I reach March 9th and she's defending a day after. So she's very nervous. My flight should not get delayed and all that, So she did this study. we went to a conference, and at a conference I met somebody from FDA. They were mentioning about this study, which we found was very interesting. So what the study is, if you see that cartoon there, you on agar plate, you put a rabbit skin, and through the rabbit skin you put a catheter. Now one side of the catheter you put some bacteria, and now you wait for 24 to 72 hours to see how much infection is going to the other side of the skin, mimicking our condition, right? If we have a catheter inserted in the body and some of the catheter is hanging out, we have this interface where skin catheter is going through the skin, and that's a very dangerous spot for bacteria to grow. So we wanted to mimic that in ex vivo skin study, where we have a catheter, here is a skin, you infect this side, and bacteria is traveling inside the body kind of through the skin. So if you see, the top one is control, and see how much bacteria is traveling on the other side. But this material, our material, is fighting and not letting the bacteria travel to the other side of the skin. So this was a nice, interesting study. Then I also talked that when we do the catheter study, we cannot just put the catheter in and do infection also, because if everything was done in a sterile environment, there is a good chance you won't see infection in a lot of catheters. I mean, chances of infection is, let's say, 5%, 10%, but then are you ready to do 100 catheters and hope 10 catheters will be infected and you can get your data? So better what we do is we actually infect the catheters before we implant them. Okay, so we dip the catheter for 2 hours in Staph aureus solution, and then we implant the catheters in the animal. Then we look after certain number of days how much is infection, because now we know all the catheters have infection. So we are actually making a very tough study for ourselves, that we are putting infection knowingly and seeing if our material can fight that infection. Okay, so now, and the beauty is we can get clotting data and we can get infection data, both. So we saw almost 70% reduction in clot formation, and see, that's the vessel we took the catheter out from. And for 28 days, so we took some catheters out at 14 days and took some catheters out. Some rabbits were 14 days, some rabbits were 28 days. And we saw 50% reduction in infection or clotting for 28-day data. This was a really good data. I mean, I almost didn't believe in it, but we did N of 4 or N of 5. So there are 4 control catheters, 4 are linaural catheters. So basically what it's doing is, when the catheter was inserted, we took pieces from both ends of the catheter, the one that was near the skin and then one that was closer to the heart, and the tissue also, and we homogenized it and looked for infection. 14 days we saw almost no infection on one of the ends and some infection on proximal end and some infection in tissue. But for 28 days, almost no infection anywhere. So over 28-day period, our catheters fought that infection and just killed all the bacteria. This was a very, very exciting study for us and very exciting data, that you are actually eliminating all the infection of a catheter. But, and you can see control had lot of infection. So that means that infection worked. All the control catheters were infected. So infection is working just fine. And the standard deviation was so tight that it was done very, very reproducibly. So this was, and we have not published it yet. Hopefully we are going to submit it in next 1 month or so. So this is the story with this type of study. We have already talked about this. This video won't work, so I'll play a different video for you later. Whoa, oh, it's working. Wow. Okay. Wait. Okay. I didn't know it'll not work. It'll work. Actually, it never works on my computer. Okay, so let's do one thing. This is an example of we developed a super hydrophobic coating. Okay, that means the contact angle is greater than 150 degrees. This paper is published. I don't know. I didn't put it here because I thought the video will not work, but I can send it to you guys. So what we did when we developed that coating, of course we can coat tubes and catheters and everything, but we thought to make the study really tough, let's coat sponges, you know, and you can imagine sponges, like if you put liquid, it's going to just grab it, like, you know, and that can be blood or it can be bacteria. It's so absorbing. So here is a control. That's what you expect, right? A sponge should be. Now, this is our treated sponge with super hydrophobic material. So you can imagine if this sponge is, this is a coating, you are repelling everything, right? And here is another example. And imagine we did this study with some dye or something, but we can work with blood or whatever. This is a control, a sponge. So the idea is, how can we make the material so hydrophobic that it's repelling everything? Then now I have like some 15 slides or something talking about different projects we have been working on. I wanted to show more, but I couldn't make more slides. The goal of my lab is more or less medical device, infection, clotting, in whatever applications we can go for. We have even published a paper on packaging materials, prevent infection in packaging, you know, just wherever we can apply that. You know, we have make sutures, fibers, hydrogels, coatings, films, tissue scaffolds, all these different applications. Okay, so here's an example. I have a good friend, Joe Potke. He works for US Department of Veteran Affairs. We are working together for 12 years. He's an expert in making microfluidic artificial lungs. Okay, so the idea is people waiting for lung transplant can have this artificial lung that they can carry, so that they don't have to be hospitalized and wait for a lung transplant. So here is an example of one of his microfluidic lungs, and you can imagine the size of the channels and small. So our role is, how can we prevent clotting and infection of this lung? You know, the channels are so tiny, it's so easy for these channels to clot, and then your lung will stop working if these channels start getting clotted, right? Again, just keep in mind, the beauty is, I cannot do the things that he can do. I cannot make this artificial lung, and he cannot do the things that I can do. So you have to learn to collaborate, and you have to rely on people from different backgrounds. Another idea, so I have this a friend and a collaborator, Dr. Andriy Baczynski. He worked for Army. Now, last 2 years, he's not working for Army hospital anymore. But their idea is, if a soldier is badly injured on battlefield and they are badly bleeding, how can we keep them alive when their kidneys are failing, lungs are failing, heart is failing? How can we have extracorporeal devices that can keep that soldier alive for 2, 3, 4 days before they can be brought to a proper hospital setting? You can imagine, we cannot give heparin to this soldier. because they are already bleeding. And if you give heparin, they'll just bleed much faster. So how can we make surfaces that will prevent clotting and infection but still keep the soldier alive? Another area of research that we are working on is instant clotting. So our company has a grant from US Army for this work. Instant clotting and bactericidal faster healing wound dressing. A soldier is badly injured in the battlefield and bleeding, actually bleeding so badly that they are not even concerned about infection. They are just concerned how to stop the bleeding so that they won't bleed to death. But at the same time, this type of environment in a battlefield, there is a lot of infection also. So how to take care of infection and prevent too much bleeding from happening with the wound dressing. So we are doing some work with the University of Miami, the company, and we are testing our wound dressings in a pig model there to see, can we clot, can we prevent infection of these wounds. Next, so these were my 3 PhD students, Megan, Jitin and Arnab. And this is that NO generation technology coming back. I forgot to delete that slide earlier when I was saying. So we have published these different papers with selenium or copper in animal model. I think one of them, yes, this one was in animal model. The idea is we have a NO releasing polymer and we can put a NO generating catalyst on top, so that the NO releasing polymer is sending NO donor, or sorry, not NO donor, only NO gas, and the top coat has the catalyst in it, selenium, copper, and when blood is coming in contact with that, it's generating NO. Now you are getting more nitric oxide release because you have a combination of NO release and NO generation both happening at the same time. The reason is, over the years while doing the studies, we also little bit found out, I'm not 100% sure, but we think the blood does not have enough packets of that RSNO, that S-nitrosothiol, that can get converted to NO when it comes in contact with the catalyst. So if there are limited number of packets in blood that can get converted to NO, then NO generation can sound very fascinating because there is no reservoir. There is no limited amount of NO donor. You can have the catalyst and it can keep generating NO. We think the amount won't be enough either to have enough NO or to go for a very long time. So the idea is, immediately after the surgery, when the chances of infection are high, you have NO donor and NO generation both working together. And now after some time, even if one of the technologies are not working completely, the other is still going. But for this type of work, now you can imagine, okay, if you have to design a coating that is NO release plus NO generation, what will you think about? Like, what are the factors you will have to keep in mind simultaneously? Yeah, we want both of them to happen. Yeah.
We cannot coat our NO releasing material on the surface. It has to be inside.
Very good. Yes. The top coat cannot be releasing. Okay. And why?
Because the NO gel won't work.
It won't work, right? Because it needs to touch that surface. Okay.
And the NO surface should be penetrative in it enough so that NO release is not hindered by that.
That mostly we don't worry. I mean, NO can easily escape. What else?
So the top surface should not be reactive with RSN. Just act like a available surface just for the conduction of that reaction and not exactly bind with it in another fashion.
difference between generating and releasing? Generating means it is.
Okay, I'll tell again. It's a little confusing. So NO releasing is you have a NO donor compound like SNAP, diazonium dilate that has NO bound to it and it's releasing it. We are putting that we are synthesizing it and putting it in the polymer. That's NO releasing. NO generation is we are only putting catalyst, copper or selenium, let's say, and we are relying on the packets of RSNO that are floating in blood, that when they will come in contact with that catalyst, they will release NO that they were carrying in their packets. We are not putting a NO donor ourselves. We are relying on the NO donors packets present in blood. So it's just converting its packet to NO. So that's called NO generation. We call it NO generation. It's generating NO. We have published papers that are just NO generation also, where we didn't put any NO donor. Forget this green coating. We just put catalyst, put it in animal, and the blood in the animal is generating NO. But then we thought, let's go a step further and combine NO release and NO generation in the same coating. And that's what this was. But there is one more challenge here that you are not talking about. Who can tell me that?
Chain surface protein that is going to bind on that surface. Bind. Because a protein, if the surface is very clogged up with protein,
then we won't be able to do the generation. So very interesting. Vince, his paper is not published yet, but he has all the data now where he's making NO generating slippery surface. Now that slippery layer will prevent proteins to stick to the surface, so it can keep generating NO. Okay, that's a good thought. But there's another challenge in this that you guys are not talking.
Snap needs to pass through that top material that is.
Yeah, I think he said or you said before, and that's not a worry. That's fine. We can easily, NO can easily pass. So why, oh, you said SNAP needs to pass. Why did you say SNAP needs to pass?
Because Snap is releasing that NO. Not Snap needs to pass, but the NO from Snap needs to be released.
Yes, and that's fine. That's easy. That's not a problem. Yes.
The combined release of NO gen and NO releasing substance of the combined NO release should be optimal, that it doesn't induce any toxicity.
Sure. The levels should not be very high.
My doubt, because generation and that's why I asked you that both the levels, or if they're releasing and generation the same process.
Oh, so NO release and generation does not need to be same. I mean, maybe initially NO release might be 3 flux, NO generation might be 1 flux. Over time, NO release might become 0.5 flux because NO is finishing, SNAP is finishing, and NO generation can still stay at 1 flux. The goal is their combination is good enough to do the job. So that's fine. But there is one thing that you guys have not thought. Take 1 minute to think about it.
Both the systems active together or in conjugation.
Oh, that's a good idea. Now, it can be very nice if we can on and off systems, but the problem is we cannot. As soon as I put it in the body in moisture and 37 degrees and all that stuff, how can I stop NO release not to happen? Or how can, see, if I can stop it, then I can be like, okay, for this time it can get on, this time it can get off. We can do that if it's external wound dressing. Then I can do is shine a light, so you can have lot of NO release. Stop the light, your NO release is there, but very low numbers.
So it will already happen, like if during the day we are working, playing, or doing whatever, we're exposed to light. When we go to sleep, usually people sleep with dim or no light.
Yeah.
That can be conserved anyway.
Yeah, but now I'm just saying this is an implanted device. So one thing that you guys are not thinking is let me forget this picture. What will happen if I have a film that has NO releasing polymer, let's say snap, and in the same polymer I add copper. So I have copper or selenium plus snap in the same polymer. Okay, and then what will happen if they react? No. Why not a good thing? What do you think will happen?
Whatever some force will be generated into that, and that can induce protein.
So the NOs themselves will start reacting with the copper selenium inside and not be.
No. So when I talked about that snap slide, I said the trigger to release NO is heat, light, and metal ions.
So it will start releasing a lot of.
So see, snap is one type of RSNO. It's synthetic. We made it. RSNO in blood is also that family, but it's more endogenous. So if we put snap and copper in the same coating, whatever the packet was reacting with copper, now that is, we are putting both of them together in a coating, they can start releasing lot of nitric oxide very fast, because NO snap is breaking to release NO, but copper is causing it more catalytically release a lot more NO, and now more NO is releasing. It can be toxic or may not be toxic, but we might lose all the NO very fast, because all the snap is getting consumed fast because of copper. So when we did this study, I don't remember, it has been few years, whatever, 2018, 2019, I don't know what years. We found out that NO levels were very high. So we put a sandwich layer between snap and copper so that they don't interact with each other. Now NO is coming out and copper is interacting with the packets in blood. It's not interacting with the snap in my coating. When we were not putting this sandwich layer, copper and snap were touching each other, and now we were releasing a lot of nitric oxide. Actually, it was so much that even while making the film and drying process, we were losing some of it just in the drying process. Ooh, I don't remember, but we can just make whatever. I mean, it's up to us, right? Like whatever the requirement is. So for catheters, if you want a catheter to be 2 millimeter diameter, outer diameter, we can have a total inner diameter of, let's say, 1 millimeter, outer diameter is 2 millimeters, so we can have that thick, that is our working thickness, right? Here, if we have a coating of a tubing, we can make 100 micron thick coating. It just depends on the application, how much room you have for thick or thin coating. But yes, thinner the coating, less snap you can put in. But the beauty of that swelling method, you remember snap swelling inside the tube is impregnation method. We don't need to coat it. We can just put all the NO donor in the wall of the existing tubing and dry it, and all the solvent is gone, but all the snap is trapped inside the thickness of the wall. So that's the beauty of that method, that you don't have to worry about thickness.
Something if we like any mesh-like surface, if we electroplate it with whatever desired material we have, okay, and then we coat it with something that is preventing the snap to react with the metal ions, so we can create a conjugate layer, which will be even more thinner. I'm not sure that it will be thinner than this or not, but
completely understand.
Let's say you were talking about whatever a polymer if we impregnate it with snap and everything. So we are just putting the snap in that. So what if we take the polymer, we electroplate it, like it would be a very thin layer of whatever metal that we are doing. We electroplate it, put a barrier layer like this in the mesh itself or in the polymer itself, and then impregnate it with snap. So this might reduce the thickness.
By reducing the thickness of the copper electroplated layer.
Maybe of the copper electroplated layer and the overall thickness of our material, whatever we are trying to.
But see, we have to also remember, if you are coating a tubing inside of a tubing, can you do this method inside the tubing, right? Let's say you have a 1 meter long tubing and you have to coat inside of that with that method. Can we do all that? And how much will be the cost of that method? So, I mean, for a publication, maybe yes, great idea, we can try it. But can it become a practical solution? Those things will have to be figured out.
Related to what this question was, have you ever studied to what extent that this are whatever NO generating material, what is the depletion rate of whatever metal that we are using?
Yeah, so usually it's not rate limit. Usually catalyst is not a problem for us. It will keep going. But do we have enough packets in blood to keep going? That's a more of a concern than our catalyst. We already talked about this. Then, this study is, so Akram was a postdoc in our lab and he did a lot of good work. So he did his PhD at University College London and he was very good at making super hydrophobic surfaces. So this work, like you can imagine, see the contact angle is 154 degrees on his surfaces. So this is a silicon rubber and after he adds his coating to it, it makes the surface super hydrophobic. And he also did is, he actually made this surface silver and nitric oxide releasing both. So, you know, silver is a standard used in industry right now. So we thought, why not do a surface that's like combination of silver and nitric oxide and super hydrophobic. So it's super hydrophobic, silver releasing, NO releasing surface. And we did a lot of studies. It's a good journal. And we saw that combination was not cytotoxic. Last graph, we were able to reduce infection. Combination of everything, silver, NO, super hydrophobic, was better than anything by itself. And it also prevented clot formation on the surface because of super hydrophobic and nitric oxide. Not because of silver, because silver has nothing to do with clotting, but super hydrophobicity and NO prevented clot formation. Let me play some videos for you. So again, some of all these videos are Akram's work from different papers, some sponges, some tubings and all that. So video number 4, let's look at it again. Polyurethane, these two, this is a control and all others are coatings of different type. And I think he had a water droplet, coffee droplet, and I don't know, he had different like Coke. Of course, we did it with blood also. So he had different types of solvents that he can repel a lot of different things. Now let's do number 13, the white, this one, the white sponge. Now this is just a sponge, a regular sponge, right? Then number 13, the gray sponge. Now it's a coated sponge with our material. Oh, see, the video is so fast because the blood just goes. And imagine sponge is so difficult for things to just. Okay, now let's watch a tubing video. S8. Now this is a regular tubing in blood. We are just dipping it for whatever time, and very fast blood is starting to stick on the surface, right? Now this is our coatings that we have put on. This is no nitric oxide, just totally clean. Different types of tubings, different treatment equally. Yeah. So they all are coatings that are, yeah. So the controls were this video. Some are slippery, some have super hydrophobicity. They are just different combination. This is, so sometimes, see, if you want things to grow on it and stick to it, and if it's so repelling and it's a permanent implant that's going to just stay there, now that can be an issue. Okay, now video number 14. So again, same sponge with some blood study, right? This is all blood on it. This is treated sponge. So these were some Akram's videos. We also talked about this work where we are trying different levels of bacteria and with time how NO can fight to certain level very well, and then it still fights but not at 100% or something. Okay, Vijay was a postdoc in our group and he had lot of experience with virus bacteriophage type of work. So this work was NO plus bacteriophage technology. That how we can act, NO is killing, bacteriophage is killing, and how their combination can be very effective. Then Dr. Chad Schmidt is my very good friend. Actually, last 5 days he was in India. He went to a conference in Jaipur, but we couldn't meet. I mean, he's in my university, so I see him all the time. But meeting in India would have been special.
Last time you said Jaipur temperature.
Yes. So, and Ryan did his PhD with me. So this paper was NO release plus heparin combination. Heparin is also a standard. So can we combine NO with heparin and show their synergistic effect? So that was the idea here. A lot of time we want to do this type of studies because convincing clinicians to just go away from the standard might be more difficult initially. But if we say, okay, you use silver catheters, that's great. How about NO plus silver is better than silver alone? Or how about NO plus heparin, it's better than heparin alone? So we did some rabbit studies with that. Then Arpita is a postdoc in our group. She did her PhD from IIT Guwahati. And this was her paper, making liquid-like omniphobic solid slippery surface, which is super anti-biofouling. You can even see on that image, there is a lot of biofilm growth on the top, Staph aureus and E. coli, and our surface had basically no biofilm growth. So this was that omniphobic anti-biofouling surface. Then this is another interesting work. We had a grant from NIH, we have a grant from NIH, and we have submitted a renewal. This work is with Dr. Chris Sidlicki, who's a professor at Penn State, and Elaine is a student who's defending on March 11th. So his technology is making these pillars on the surface inspired by shark skin, so that if bacteria wants to sit on the surface of these pillars, it's very difficult for bacteria to properly adhere to the surface. But then we went a step ahead and we thought, why don't we make these pillars NO releasing? So we have these NO releasing pillars. And in this work, we did some animal studies and we did in vitro studies. In vitro study was, we had these blood bags, and his group did this work, we did animal work. And we had pump, we had catheter sitting in the blood, and then over time we saw how textured really looked better than control, and NO releasing textured looked even better than textured. And same for animal work, the combination of textured or NO releasing pillars were much better than NO release or pillars by themselves. Approvals to do blood studies with animal blood is much easier. Human blood is also okay, it's just more process and more approvals needed. So it's a little bit more complicated than animal blood. Yeah, something, because of course, whenever humans are like, it's more protected, right? Animals also, animal studies are more protected, but if you're drawing animal blood, that's little less harmful to animals. So that's why it's a little bit more easy. The next study, Priya did her PhD in our group 4 or 5 years ago, and Dr. Lachlan is my very good friend and collaborator. We have published 5-6 papers together. So this work was NO releasing zwitterionic surfaces. So zwitterions are hydrophilic surfaces, and you can see that, you might not see it very clearly because these catheters were so small. We measured the contact angle outside the catheter and inside the catheter with zwitterion. So the contact angle is much less, like maybe 40 degrees contact angle with zwitterion. And we did an animal study and we showed that how ZNO or zwitterion plus NO surface had much less clot formation compared to control. Like control looks terrible, right? And NO looks good, zwitterion looks good, but the combination looked even better. Megan did this paper on NO releasing urinary catheters. So we bought urinary catheters, we made them NO releasing by our method of swelling snap into the walls of the catheters, and did bacteria study, toxicity study, and everything. I'm just like, my goal is to just give you big picture. If something is really exciting to you, you can go find the paper, read more about it. And honestly, if the papers were published 3, 4 years ago, I don't even remember the nitty-gritty details on the paper. Okay, this was also good work. Mark did PhD, and now he's a faculty member, a research faculty member in our department. NO releasing hydrogels. So these hydrogels were fluoride releasing also, and the goal was preventing infection, and can we prevent demineralization of the enamel. So this study was very cool. We did a lot of bacteria study to show that with this was pluronic hydrogel, alginate hydrogel with fluoride ions in it. So what we did is we treated the hydroxyapatite discs with our material, and then we put demineralization solution in it, and the one that was not treated with our solution, this happened, all these pores started forming, and the one that was treated with our solution, it had strengthened the enamel so it does not get that demineralization happening on it. Next study, Morgan was also a PhD student. She did that catheter lock solution study. You lock the catheter with the solution. Now, what's the benefit of nitric oxide releasing lock solution? Yes, but most lock solutions are either heparin or antibiotic. They are either you're fighting clotting or you're fighting infection. Our lock solution is fighting clotting and infection both. So this was that type of study. Sama also did PhD. She worked a lot on silk. We published multiple papers on silk. So she made this gelatin silk scaffold, NO releasing. Basically, this was her gel. It helped in antimicrobial properties. I purposely am showing you different types of studies, that is like zone of inhibition study from those gels. And this gel was really good for tissue engineering type application because it helped in cells to adhere to the surface. And it made the gel, this network of silk made this gel very strong and not very fragile that will just break. So you made it stronger and antimicrobial and easy for cells to grow, so great for tissue engineering type applications. Dr. Melissa Reynolds, she works with nitric oxide work. I know her for 15 years now. We have published together a lot. We worked on this project, so she works a lot on MOF, metal organic frameworks. Idea is same of NO generation, but sometimes if you just add copper can leach out and cause toxicity. Metal organic framework, it's a bigger molecule and it's more secure and it does not come out as a leaching so easily while doing this NO generation thing. So that was this idea. Then almost done, few more slides, then we can talk about the exam. Nitric oxide is not very strong for antifungal applications. Okay, and you can see that in this graph, the bottom one at the corner. C. albicans, nitric oxide SNAP, it's little better, but it's not great. So this idea was, can we immobilize amphotericin B, which can take care of fungal infections, and nitric oxide can take care of bacterial infections. So now nitric oxide can prevent clotting and nitric oxide can prevent gram-negative, gram-positive infections, and amphotericin B can take care of fungal infections. Arnab did this work on antimicrobial peptides, contact killing, niacin. So if bacteria comes and touches the surface, it will kill it. So, and NO is always there to kill. So NO is killing things, but if somehow bacteria is still surviving and coming to the surface, niacin is killing it. This was another cool technology, snapicillin. So ampicillin is a common antibiotic and SNAP is a NO donor. So we covalently bound SNAP to antibiotic. So ampicillin and SNAP, and we call it snapicillin. Now the idea is antibiotics cannot penetrate biofilms, right? You need 1,000 times the dose to penetrate. But NO can penetrate biofilms. So the idea is this snapicillin, if you have a surface that's badly contaminated, you expose it to snapicillin solution. NO will penetrate antibiotics and can kill the bacteria, disperse the biofilm. I don't know if I said the right thing. I just said it right. Okay. So you have a biofilm and NO will go inside the biofilm, disperse it, and now antibiotic and NO both can kill it. But by itself, antibiotic cannot kill a biofilm. And here is that data. So what we did is we grew MRSA on different surfaces, on a polymer surface, let's say. And then we exposed it to ampicillin solution. So first of all, we looked at MRSA and we homogenized it on a catheter and we saw this much bacteria was there. Other surface we exposed to ampicillin solution for whatever time, I don't remember. And then we homogenized it and we saw so much bacteria was there. We exposed it to SNAP solution, and when we exposed it to snapicillin solution, it took care of antibiotics better than anything else. So one paper is more solution paper, snapicillin. One paper is more polymer, that has snapacillin releasing properties. NO releasing anti-fouling surface, we did a animal model on it also, the ECC 4-hour model. And you can see the contact angle. When you had only NO surface, the contact angle was high. When you had the NO releasing anti-fouling surface, contact angle was much lower, and it worked great in animal model also. The last one is NO releasing sutures. So we made PLGA NO releasing sutures, which can now fight infection and reduce infection, and over time the suture will go away anyways. So that's the idea with NO releasing. And Lauren did her PhD last year. So, and she's working for the company now. So to conclude, we did a lot of things today, but main thing is we can release nitric oxide for long periods of time, right? That was one of the challenge when I started working in the field. We can swell or impregnate sap into the walls of the materials, so we don't have to extrude. So that was another challenge. We can sterilize it. It is shelf stable, which was another challenge. NO releasing materials do not prevent protein absorption. So we can combine it with super hydrophobic material, super hydrophilic material, slippery material. So we can take care of that NO plus anti-fouling. And we are working on catheters, stents, grafts, urinary, wound dressing, endotracheal, dialysis. This is like so applicable to so many different applications. So really cool technology, can be very easily combined with antibiotics, heparin, hydrophilic, hydrophobic, whatever type of other technologies you want to combine it with, it's very easy. You can make hydrogels, or you can make sutures, or you can make films, or so very versatile technology. And with that, I want to thank a lot of people, lot of my group members who did a lot of this work. I think in last 10 years, I have not done a single experiment by myself. So of course, the credit goes to all the students who worked in the lab. I had so much more data by other people. I just ran out of time. So if my students are upset that I didn't show their data, I'm sorry. And of course, thankful to so many collaborators. They all have so much different skills that we don't have. Funding agencies, if you don't have funds, you cannot do anything. So yeah, so this is what approximately what our lab does. So exam tomorrow. Let's talk about that. So any questions for exams? Yeah, for exam. So exam will be 11 to 1 tomorrow. And my understanding is if you don't take the exam, you don't get something. You still get your certificate, but you don't get your credit. It's a 2-credit course or 1-credit course, whatever it is. You won't get that credit so you won't get your credit if you don't do the exam. First, for online reasons, let's little bit talk about big picture of the work I talked without talking nitty-gritty details. Do you have any questions? Yeah, but I mean, if you have some other questions or any ideas or anything, what you think? Yeah.
Inside the tube. Use it inside the tube.
So the work there, that's interesting question actually. It's interesting question. So what they do is they make that sheet and I think they roll it into a catheter. Making it inside a small catheter is very challenging. That was your question, right? How they make?
Was it pre-manufactured or did you fabricate it?
No, so I think they make it into a film and then they roll it into a catheter, because going inside a 2 millimeter diameter catheter and making those is, yeah, how do you do that? So see, that's a good example of how do we do commercialization with that type of material, right? So that's a little challenge that we might face with that type of technology.
But you were fabricating, you could have casted it on something like a pole or anything that has all these grooves and you.
Yeah, but see, I don't remember exactly, but I think those pillars are half a micron apart. You know, I think so. Because if the size of a bacteria is 1 to 2 micron, those pillars are maybe 700 nanometer or something like that. So they are like so close, making it in a tiny lumen of a catheter can be really challenging. So if I ask you, out of all the stuff I showed you, okay, no, you all have answered separately. All the different types of things I showed you, what was most exciting for each one of you? Like, you know, I showed so many different papers. This is Dysanium dilate, SNAP, Zwitterion plus NO, this, that, suture. What technology was
Really nice. It was really innovative to use two materials, like one which was generating NO and one which was.
so NO release plus NO generation was you liked a lot.
I got something in my mind which I'd like to share.
Okay, okay,
But yes, it can be something.
okay,
It is.
okay, okay.
I love Snappicity one because the next pandemic. And so, as I don't know about USA, but in India, antibiotics are so available over the counter, and we're just taking it like candies.
Yeah,
So, and we're not even completing the dosage.
yeah,
So, that could be the next thing. And we can obviously foresee just like pandemic, like COVID or the antimicrobial pandemic that's up here. So we need some technology to curb it. So I really love the idea and the idea that NO being a gas can penetrate that wall, whereas 1,000 times more of the antibiotic dosage could not pass the filter. It's
yes.
very efficient, very doable, and I believe when the need comes, it could be scaled up to a larger extent.
Okay, so one person liked Norel plus Nogen, one person liked Snapicillin. We have a grant from NIH for Snapicillin, and actually we have not published and we have not filed for patents, but we are making different combinations.
Yes.
So we are making that similar style NO antibiotic, different types of those. Okay, do you have any that you particularly liked? Hydrophobic? Yes. Okay. Anybody? See, everybody does not need to answer, but if you feel something you really liked, then you can tell me. Either you didn't like anything or you liked everything, and that's fine. Anything that jumped out? You mean where we put SNAP into the walls of the
Yes.
tubing? Okay. So diffusion method to impregnate the tubing. So see, that's a very practical. I mean, all of what you are saying is useful, right? I mean, I did it, so I think it's useful. That's fine. But see, Snapicillin has one type of advantage. Norel Nogen is different. But yes, you have both technologies working together. Your style, what you are talking about is it's very helpful for manufacturing side of things. It makes it easy to manufacture. Another good one that I really like is this Linorel, liquid infused nitric oxide releasing surface, and we have trademarked that name also for our catheter technology because you're making the surface anti-fouling and antimicrobial and hemocompatible, everything with that technology. And that actually the animal data showing all the bacteria dying on the catheter, I really, that's like, I like that a lot actually.
So artificial skin,
Yeah.
that is also very appointing, and I think
So the thing is, we have not pursued too much of that. You know, lot of times this research is very grants driven also. The area where we have got grants or the area we feel we can get future grants, we lot of times want to go in those directions.
I mean that is very interesting.
No, I know. I like it too, but Another thing is, if you don't have funds, doing animal studies is very difficult because you cannot use one grant money to do a different animal, right? So a lot of times students who want to do studies, they want to do catheter or ECC type work because we have funds to do those type of animals. But if a student develops a really nice artificial skin, we don't have funds to test it on animals. We can write proposals, but that is our 2-3 year project. And if you're lucky, you might get funding. So I would love to do that more, but so far we have not been able to. So we can talk about the exam quickly. I have the paper ready. I think it will be a 2-hour exam. And 15 multiple choice questions, 5 short answer questions, 2 numerical problems. We talked about polymers, ceramics. We talked about protein adsorption, platelet activation, bacterial infections. We talked about 3 different types of chemicals: stress strain, molecular weight of polymers, weight average, number average, and that bacteria, like the generation time of bacteria. It will be a closed book exam, so you need to remember all the formula and no cheat sheet or anything for formula. You need to remember those. Actually, yeah. So it's just, I would like steps because when you are sitting next to each other, writing an answer is much easier. If your answer is wrong, you will get a 0. But if you show steps, I will give you some points for steps also. What if you made a mistake in the last step? Then you might only get 10% points deducted. So always show steps. Calculator is allowed. Phones are not allowed, whatever. Yeah, I mean, yes, because you see, our work is such that you can easily Google stuff, right? So, yeah. So, I'm trying to understand it for 10 years, and even I have not understood it. So, you're okay.
In tomorrow's exam, to ask any question from that, I actually have to understand some part of that.
Okay. So, let's talk about it a little bit. So, there is extrinsic and intrinsic pathway for coagulation, right? And you can remember extrinsic is injury. So if there is an injury, platelets are important to immediately plug that hole. If it's an intrinsic pathway, protein adsorption happens first and then platelets are sticking to it. Both pathways lead to a common pathway where factor 10 gets converted to activated factor 10, which helps convert fibrinogen to fibrin, which results in cross-linking of fibrin and forming a mesh. Even if you don't, I'm not going to ask you that cascade, I can tell you it. But there is a factor 11 that gets activated on one side, factor 7 gets activated on other side. So those are the factors that get activated. Injury side is tissue factor 7. Yes. So they start and then they come to common pathway. Common pathway releases chemicals that keep generating more and more, creating more factors, right? Another thing you should know is how injury gets plugged. So if somebody gets a cut, collagen gets exposed, activate there, and they activate and bind to each other. Fibrinogen comes, acts as a glue between platelets. Fibrin forms, cross-links, results in a clot formation. That's how bleeding stops. Or I think if you know this much, you're fine. I'm not going to ask you the whole. If you tell me to draw a whole coagulation cascade, I cannot either, right? Okay, guys. So thank you. I'll still be here. We can talk, but session is good, and we'll see you tomorrow for exam at 11 o'clock, and then we will do some class at 2 o'clock tomorrow. So, okay, thank you.